much for the invitation of Honorable Dean, Professor Hidayat. Good morning, ladies and gentlemen.
Morning.
Firstly, I appreciate Dr. Utama and University to giving a chance to do this and have a small lecture here today. Although this have many problems with my presentation. Okay, so let me introduce to you where I am and who I am. Here, your university is right here when I see the map, and I'm up here, South Korea, and Seoul, which is capital city of South Korea, located here, and Seoul National University is right here. If I see the Seoul National University is top and the biggest university in South Korea. Our university ranked 36 of QS World University Ranking in 2022, even though this is not really important. But if I something to give you information, this is the best and easy way. Seoul National University is a multi-campus university, but most of academic and research activities are doing in Gwanak campus. Here is really a big and crowded with many buildings right here. Yeongan campus is prepared especially for medical school, and they have a big hospital next to the school. And Pyeongchang University, the Pyeongchang campus is located in Gangwon area of Korea. And here we have research facilities, including research farms, animal farms, with the Department of International Agricultural Science and Technology. And recently, Siheung campus has been built for advanced future technology. And this is College of Agriculture and Life Science in Gwanak campus. Okay, let me introduce our lab. I'm Chorun Jo. I'm responsible for operating Animal Origin Food Science Laboratory, major in Animal Science Biotechnology, Department of Agriculture Biotechnology, and College of Agriculture and Life Sciences, Seoul National University. Our lab presently includes 2 research professors, 1 postdoc fellows, 5 PhD students, and 8 MS students, and 1 administrative staff. If you have any interest, please visit the SNU homepage. The major research interest area is ranged from development of high quality and value-added meat products, and improvement of safety, animal origin foods, bioactive compounds from natural animal resources and their application, and the muscle biology, cellular muscle production system, so-called cultured meat. So, okay, now let's go back to focus our subject to cold plasma technology for today's presentation. Well, effective method to prevent the microbiological contamination in meat and meat products throughout the life cycle is most important issues for consumer trust and safety. The cold plasma is rapid and effective non-thermal technology that can be applied for food products. Today, I'm going to introduce parts of our research mainly focused on innovative processed meat manufacturing using cold plasma technology. So first of all, we have to take a question, what is plasma? The plasma is the 4th state of matter after solid, liquid, and gas. When solid ice absorbs energy, it turns into liquid. And when water receives energy, then turn into the gas. And after the gas, we can say plasma with whole ionized elements in gas. Actually, over 99% of universe is plasma state. We can also create this plasma field in the gas state. Then the molecules in the gas ionized, and all elements turning to neutral and ionized gas, which is the plasma state. When the plasma is generated in atmospheric pressure state, we call this atmospheric pressure plasma, or APP, or cold plasma, because we can control temperature at room temperature. It contains UV photons, reactive species, and charged particles. And this cold plasma has also a flexible operation system, and cost is very feasible compared to other technology. Recent cold plasma research trend is getting more and more interest from food and agriculture sectors. In addition to the conventional use of plasma, such as polymers, textiles, electronics, and semiconductors. It estimated that cold plasma market worth $3.17 billion by 2024. Our lab divide the streams of research of cold plasma for 3 different ways. First, plasma pasteurization for food safety enhancement. Second, innovative curing process for processed meat manufacturing. And lastly, development of nitrate-rich natural ingredients with possess high biological activities, originally or induced by plasma. First, I'm going to talk about safety enhancement briefly. It is now confirmed that cold plasma technology improves the microbiological safety in the raw meat products, food packaging, and materials. This is one of our early work. We developed this flexible thin layer DVD plasma. Plasma can be generated within the package when electricity is on. The concept of our study is the application of this kind of package. We found quite good performance in inactivation of salmonella. In addition, we found the effectiveness of post-treatment duration. If we set the time of plasma for 30 seconds, then we have this 3 log reduction. But still, we have another 4 logs of bacteria which can be gone after 3 minutes of waiting without open the packaging. We call this post-treatment duration or post-treatment effect. In this study, we prepared E. coli 0157H7 biofilm on stainless steel coupon at 37 degrees Celsius for 7 days. Then we treated plasma in different solutions that are usually used in food industry, including deionized water, sodium chloride, or sodium hypochlorite. Then we found the reduction efficiency was the best in sodium hypochlorite with plasma technology. Then to know what are the main chemical species on this inactivation of E. coli 0157H7 biofilm, we detected the levels of nitrite or nitrate ions and hydrogen peroxide, and peroxynitrite level, and hydroxy radical levels. From this study, it could be summarized that hydroxy radicals and peroxynitrite were the major chemical species for inactivation of this E. coli 0157H7 biofilm on stainless steel coupons in this particular environment. This is plasma activated droplet or mist type plasma generating system. This is a prototype of our laboratory, and it works like this. Okay, when water passes through here, plasma is generated within here, and reactive species are dissolved in the water, and this water can be plasma activated water, which have effective in killing bacteria. After treatment, the plasma activated droplet transmittance electron microscopy revealed that the membrane of cells were severely damaged, and cytoplasms were leaked out from the cell internal cell, And that is the reason of the cell death. The action of cold plasma on bacterial cell structures are various. The nature of the proteins or enzymes, oxidize lipids and fatty acids, damage the nucleic acid, DNA or RNA, lesions and perforation in cell membrane, and breaking the chemical bonds in cellular structure. If you want to study further, please see this review paper, which is very useful. Now let's go into the innovative plasma curing process. During the lab work in microwave inactivation by plasma, we found a slight color change in meat surface. This color change is not very significant, but our laboratory members see meat surface every day, so quite sensitive than other people. So our students recognize the slight changes in meat color after plasma treatment. Myoglobin, as you all know, is a major compound determining meat color. The amount of chemical status of myoglobin affects meat color. For example, beef is darker than pork, mainly due to the higher concentration of myoglobin. Even if you have the same meat, but the color of meat could be different in a vacuum pack and wrapped pack because of the chemical status. Myoglobin is different. So myoglobin is composed of globin protein with the heme ring here in the center. Heme group is also can be divided into protoporphyrin and ion in the center. When you use this photo, you can see that general color myoglobin dissolved in water or phosphate buffer by plasma treatment time. So from the literature, we found that there are 5 kinds of myoglobin forms to make meat color green: sulfamyoglobin, chylomyoglobin, bodoheme, nitrohemine, and nitrimyoglobin. And sulfamyoglobin and chylomyoglobin are usually found by microbiological action. They have a globin protein without any deformation of globin. Bodoheme and nitrohemine does not have a globin protein in myoglobin. At first, we cannot find the specific UV spectra for sulfamyoglobin or chylomyoglobin because they have their own characteristic UV spectra. Thus, they are not one we are looking for. Secondly, the meat color A value, this meat color A value, redness, was changed from protoporphyrin ring structure. So we have an idea that the color change maybe come from the protoporphyrin ring structure instead of globin protein. In addition, when the plasma treatment time increased, there is no changes in CD, circular dichroism spectra, which shows us that there is no change in secondary structure of the protein in myoglobin. In addition, we cannot see any changes in molecular weight after plasma treatment for myoglobin. Therefore, the globin protein in myoglobin is intact after plasma treatment. It means it is not bodoheme or nitrohemine. So now we have nitrimyoglobin left. We found that when the met myoglobin exposed to nitrite-rich condition, it goes to nitrimyoglobin and take green color. So when we see the concentration of nitrite ion in environment of plasma, we can see the increase of nitrite content in the environment after treatment of the plasma. So after many experimental trials, we proposed the mechanism of green discoloration of meat by plasma like this. When the plasma is treated, plasma is treated to meet surface, production of nitrite or nitrate ions occur in aerobic condition. Then it goes to myoglobin, which shows green of the mid surface. And with this study, we also found that we can provide a strong reducing environment. Then this myoglobin with nitric oxide goes to nitrosamyoglobin, which shows a beautiful red color. This desirable pinkish red color is the color that we ordinarily see in processed meat products, like sausage or ham. So we did another experiment based on this hypothesis. Here we have a control pork, and only plasma was treated on pork, and C was immersed in 0.5% ascorbic acid solution for 10 minutes to provide the strong reducing environment. And D was C with 20 minutes of plasma treatment. We thought this, as I said, ascorbic acid may provide reducing environment, and in reality, during plasma treatment, nitric oxide bound with myoglobin of pork and show this beautiful red color without using any coloring materials or sodium nitrite and anything. During beef or pork jerky manufacturing, sodium nitrite is used for several important reasons. One of them is color development. Now here we have nitrite-free brine solution with pork, and plasma was treated. When nitrite is added, you can find this ordinary color of the pork jerky. If you compare with control, the color is not so good. You could understand the importance of the meat color in your appetite. And plasma treatment showed similar color to the sodium nitrite treated, and objective color value, which is A value, shows that the color value is even with the sodium nitrite when 40 minutes of plasma treatment time. With this color development, we could additional effect in using plasma, as we already talked, pathogen inactivation. In this particular experiment, we used the Staphylococcus aureus and Bacillus cereus, because Staphylococcus aureus is present everywhere with a strong environmental resistance, and Bacillus cereus is a heat-resistant pathogen. The manufacturing temperature of pork jerky was over 50 degrees Celsius for a drying process, because during this temperature, E. coli, Salmonella, and other famous pathogens could be killed. So we use these Staphylococcus aureus and Bacillus cereus for this particular experiment. So from this study, plasma could be a safer alternative method for pork jerky manufacturing without using sodium nitrite for color development. The most important reason of adding sodium nitrite is to obtain microbiological safety. Other important functions are color development and also enhance flavor and reduce the lipid oxidation. However, consumer avoidance of synthetic nitrite make industry think different. So they develop natural curing process. In this process, vegetables contain nitrate ions were used. Originally, many vegetables contains nitrate ions, and incubate this with the microorganism. And these microorganisms convert the nitrate ions to nitrite ion. In this case, the synthetic nitrite was not used, but nitrite is in this product as same as before. But you can label like no synthetic nitrite legally. This strategy was successful in the market, in industry, and consumer likes these kind of products. However, in this process, natural curing process, we have many limitations. Nitrate levels in plants affected by inherent variability and harvest conditions. So it is very difficult to control the sources. And also incubation time for starter culture, and also we need starter culture too. It is costly and time consuming. And even though they have a strong bioactivity, some plants we cannot use if they have no nitrate or very small amount of nitrate ions. Understanding of previous research, we tried to use nitrate-rich plasma-treated water, as plasma produces nitrite ion and can be diffused into water, because the atmosphere contains oxygen and nitrogen. Okay, so plasma can generate nitrite ions in the water, into the water. So here, when we make sausage, emulsion type sausage, plasma treated water used instead of added water. And we can exclude the sodium nitrite in the formula. Here, we also prepared the naturally cured sausage with the celery powder. We found that the color was all the same with sodium nitrite, celery powder, plasma treated water, and different from the control, which is no treatment for the color development. And the emulsion type sausage with the plasma treated water did not show any mutagenicity and immune toxicity from in vitro and in vivo toxicity test. So this plasma treated water can be used for liquid based type ham manufacturing as a brine solution. And one of the media called this as new innovative plasma curing process for ham. Then is there a more advanced form? One step forward, inline plasma curing system was hypothesized due to chemical species in gas phase during plasma generation. The hypothesis was that plasma in air can make nitrite for curing, and radicals or a reactive species produces during plasma generation for microbial inactivation. This is one of the prototype that we, our laboratory built. This is a mixer, and we installed the plasma generator on top like this. We achieved that curing can be done during continuous mixing or chopping process using plasma. Additionally, microbiological safety was improved with this chopping process. Here, nitrate content increased by time, by plasma exposure time. Temperature was also increased, but can be only, if we see 30 minutes of plasma operation gives you 100 ppm of nitrite contents, and it just gives you 10 degrees Celsius of meat itself. So it could be feasible in meat industry. Okay, next topic is the development of nitrite-rich natural ingredients using plasma technology. From the previous different research, we could hypothesize that if we have natural plant sources, and if we apply plasma technology, then we could get three advantages: increase of nitrate content in this plant, and increased biological activity, and also improve microbiological safety. As I said again, novel natural nitrate source can be produced by plasma with higher biological activities and harmonized flavor. Here, one example that we did experiment was an ethanol extract of perilla. When we prepare the ethanol extract of perilla, the nitrate content increased in a solution as we expected by plasma treatment. Here, we found increase of antimicrobial activities, including Clostridium perfringens, Listeria monocytogenes, Salmonella, and E. coli 15787 by increase the plasma time. Here, another example. We historically used garlic or onion powder for processed meat products as spices due to their balanced flavor and biological activities. So we ground garlic or onion and plasma treated, then dried. We have impressive high nitrate content in the garlic and onion powder, which are not there before the plasma treatment. Therefore, we could use this garlic and onion as synthetic sodium nitrite-free spices to manufacture processed meat. Originally, garlic and onion did not contain sufficient nitrate to be used for natural curing. After plasma process, this special garlic or onion powder would be novel spices in the meat market. Okay, so in conclusion, plasma has a potential to be applied in food industry, including safer foods, innovative meat processing, and natural products with functions. However, there are future works remain. First thing is regulation and consumer aspects. We need overall safety evaluation and confirmation for consumer approval by national or international authorities and consumer acceptance too. For practical application, we also need to confirm a concern about quality of food, efficient scale-up process, and combination of others for better performance in microbial activity. So let's just put the recent selected further information if you are really interested in this plasma technology from our laboratory. So this is all I prepared for today. Thank you very much for listening. And I'm happy to have a question if you have.
Thank you, Professor Joe, for your presentation. Now we are moving to discussion session. So I already received some questions from the chat box from Dr. Ayu and also Makbul and perwakilan UPT Puskeswan. The two questions are relatable to the safety actually. So you already told us from your presentation about the toxicity and also mutagenicity of the products treated by plasma treated water. So what do you think in the future if we use whether plasma treated water in bottled version or maybe the functional ingredients, the spices that already treated by plasma. So the safety of them.
Well, generally, the question could be about the concern of the safety, right? Yeah, to apply for the food industry, the safety is very important in every time in any countries, any consumers. And if we understand the mechanism of this plasma and antimicrobial activity of plasma, is the reactive species or particles, charged particles, reactive species. And usually, as I said before, peroxynitrite, hydroxy radicals, those are the main radicals for in charge of microbial activation. And usually, in any process in food industry, to reduce the microorganism, they use those kind of radicals or reactive species. If you use hydroperoxide, H2O2, you can use those This is the hydroperoxide is already proven for food use, and they use this reactive species itself. And I think the mechanism of plasma treatment for microbial inactivation is really same as other treatment, other food safety enhancement treatment. Therefore, I don't think that there is any problem with safety. However, authorities and consumer need to confirmation about this safety results. So maybe we could provide so many different type of safety information, like toxicity, immune toxicity, or mutagenicity, or genotoxicity kind of thing. Then after that, authorities maybe review those results and approve that. So for my individual thing is, there is no not really harmful things happen in this treatment, but need confirmation from authorities. That's it for me.
Jadi memang tadi ada dua sih sebenarnya caranya untuk memanfaatkan plasma. Yang pertama, plasma diaplikasikan ke air, kemudian airnya digunakan untuk curing daging. Kemudian yang kedua, plasma diaplikasikan ke ingredient yang dipakai sebagai rempah, spices di produk olahan daging. Kemudian si nitrit yang terkandung di dalamnya akan bekerja selayaknya nitrit sintetik. Dan sampai saat ini memang butuh data-data ya, selain dari penelitian Prof. Joe juga dari pihak lain. Jadi semakin banyak data, maka authorities akan mudah mereview kelayakan dari teknologi ini untuk diaplikasikan ke industri daging. Dan semoga aja di Indonesia ada inisiator yang membantu kita bersama ya, dari studi dari Korea, dari negara lain, untuk membuktikan bahwa teknologi ini benar-benar efisien dan tidak beresiko yang terlalu tinggi terhadap kesehatan konsumen nantinya. The second question is from Makbul Dayan. It asks about the future usability of cold plasma. Could this method change the way we consume processed meat?
Oh yeah, that's also a very good question. Well, if we think about the sodium nitrite-free sausage or ham, processed meat products, it is very important the replacement of sodium nitrite, because sodium nitrite is very important to protect consumer from very harmful materials, very harmful pathogens. In this case, we don't use any synthetic compounds. We just use electricity, put some generate plasma, and put the nitrite ion in the water, and use this water for manufacturing processed meat. It could be very easy, and also during this plasma treatment, we can have antimicrobial activity too. So I think this innovative curing process is very good manufacturing process for the future processed meat industry.
Thank you. Jadi memang tadi seperti yang saya jelaskan sebelumnya, ada dua metode ya cara memanfaatkan plasma ini untuk pengolahan daging. Prof. Joe sudah menjelaskan prospeknya karena dia akan sangat murah, dan juga konsumen kan trennya sampai saat ini ya sukanya yang menjauh dari yang sintetik, jadi pengennya yang degenerate secara alami dan tidak beresiko gitu. Jadi ya memang sebenarnya si plasma ini sangat-sangat prospektif digunakan di level industri bahkan ya, tidak hanya kalau di rumahan mungkin ya karena kita produksinya sedikit jadi tidak terlalu berefek ke mami, tapi kalau sudah level di industri mungkin akan ada gap di sana. And the third question from Bapak Saulan Sinaga, he asks you about the application of plasma technology against the virus, nowadays virus, and this SARS-CoV-2. I think so many products already developed like air conditioner or air filter using this plasma. What do you think, Prof. Joe? How about this technology in Korea nowadays? It's become popular. Everybody buy air filter, right?
This plasma technology. For this plasma technology, still there is no legal issues there. I mean, well, we can use this technology, but later our authorities do not know what is this technology is really itself. Therefore, we don't have any approval state yet. So this is developed, the technology is developed, but it's not legally approved yet.
Okay. So for food, I think it's not legally accepted yet, but in air filter, like other applications.
Oh, yes.
Air filters. I think Korea already developed this kind of product, LG and so on.
Yeah, humidifier or those also the cleaners, and also some of air filters. Yeah, they use the plasma technology for microbial inactivation.
Yeah. And so many researchers trying to prove this plasma also can eliminate SARS-CoV-2. But
also some of vegetables or fruits can maintain the quality and also delay the putrefaction, delay the putrefaction or spoilage after using the plasma treatment in the warehouse.
So it's just like irradiation technique.
Yeah, kind of, but the irradiation is very helpful for people when they exposed.
Yeah.
Of course, it is safe process, but if it exposed to human people, it is very harmful. But plasma is not that harmful because the energy level is very low when compared to ionizing energy.
Okay, I got it. Jadi plasma ini ya seperti kita ketahui sebenarnya sudah dipakai kalau di teknologi air conditioner atau air filter, bahkan kalau di negara maju mungkin ada beberapa kendaraan mereka sudah dimasukkan teknologi plasma untuk memfilter udara, termasuk juga mengeliminate segala virus ya dan bakteri yang ada di udara. Namun untuk membuktikan yang apakah bisa juga mengeliminate SARS-CoV-2, mungkin sudah ada ya publikasinya, tapi ya ada juga mungkin perusahaan-perusahaan yang klaim itu. Dan karena Prof. Joe bidangnya di food, jadi beliau menceritakan aplikasinya di pangan, bisa dipakai di warehouse gitu ya untuk menyimpan produk-produk kering dan juga buah dan sayur.
Diki, itu African swine fever, bukan SARS-2. African swine fever.
Okay, all kind virus lah ya.
In swine meat lah, pig meat,
gitu.
Ya, saus.
Oke.
Bisa nggak mengatasi African swine fever karena 360 hari dia bisa tahan pada posisi freezer.
Tapi Tapi dia kan menginfeksi hidup kan, ternak hidup.
Iya, iya, I know.
Kemudian setelah dipotong, mau diaplikasikan, maksudnya diaplikasikan si plasma ini supaya menghilangkan itu. Oh gitu.
Dalam
Mungkin
prosesnya
ya
dia kan
beliau belum,
saus
ya,
kan tadi.
ya kemungkinan beliau belum mencobanya
Belum ya.
ke si virus atau mikroba yang mengakibatkan penyakit itu. Jadi tadi yang di presentasinya itu semua hasil penelitian beliau yang mungkin bisa dijadikan ide untuk penelitian selanjutnya.
Oke. Thank you.
Next question from Pak Andre. Does this plasma technique apply also to for raw milk pasteurization?
Oh, yeah, we had some experiment about raw milk, also milk products, and we found that well, of course, there is some effect on microbial inactivation, but the thing is, when we treated milk by plasma, the flavor changes too much. As you understand, those oxygen species, reactive oxygen species or reactive nitrogen species, give us oxidation of the fat or oxidation of the lipid, and those compounds, those reaction gives us really bad flavors sometimes. And milk is really, well, I don't think that is acceptable after the plasma treatment. Okay, so
thank you. And then I will continue this question. How about pulse electric field? So when you compare plasma and PEF to treat milk, which one is better in terms of quality?
First, electric field. Yeah, this is also one of the emerging technology. I think the effect of microbial inactivation effect, it could be same, or in my experience, well, it's my experience. I'm not very well on the electric field study. However, cold plasma is a little better than the pulsed electric fields in terms of microbial inactivation. However, the flavor change is not very, I'm not so sure, but PEF could be less flavor change than plasma.
Okay, we got it. Jadi Pak Andre yang ini dua teknik berbeda. Kalau plasma menggunakan plasma, kalau pulse electric field menggunakan listrik. Cuma si plasma ini karena dia menghasilkan radikal bebas, apalagi di susu ada lemak, itu akan lebih susceptible, ya, akan menghasilkan oksidasi yang berlebih dibandingkan dengan elektrik. Namun sisi baiknya si plasma benar-benar bisa mempasteurisasi lah, membunuh bakteria pembusuk di dalam susu, lebih baik daripada PEF. And next question. Okay, about the water used for producing water, plasma treated water. So, is there any requirement about the standard of the water quality that must be considered?
For experiment, we use deionized distilled water, but you can use tap water too. Okay. So, of course, if there is there are impurities like a lot of metal ions there, then might have some happen. But ordinary tap water or ordinary drinking water could be fine.
Oke, mungkin standar drinking water di Korea dengan Indonesia jauh beda ya. Jadi kalau misalnya ingin studi tentang di Indonesia, mangga dipersilakan Bu Sofi ya mempelajarinya. Okay, next question from Pak Doni. Digunakan pada daging babi. Oke, kalau misalnya ini contohnya daging babi karena memang lebih umum ya di Korea untuk meat products. Gusnim, have you ever tried to use this plasma technology for other meat, for example, chicken or beef or lamb?
Sure.
Is there any differences?
I don't think any difference at all. We did for microbial inactivation, we used pork, beef, or chicken all together, and they are effective. They are effective in different way. So yeah, you could use any sources, animal sources.
Okay, thank you. Next question from Mr. Rahman. To extend the shelf life of a particular product, not only the problem that we have to consider, but also. Okay, how plasma technology alleviate again the oxidation process? So do you, I think we need antioxidant to add, or maybe we just use the plasma itself.
As you all understand, any processing or any treatment have advantage and disadvantage. If you have antimicrobial activity, then you may have this oxidation kind of undesirable effect, because these antimicrobial activities come from reactive species, and this reactive species should occur some chemical reaction in lipid or others. So you can use antioxidant, or you can use any other treatment to reduce those oxidation.
Jadi memang masalahnya karena dia memproduksi tadi free radicals, jadi lebih ke oksidasi lemak dan protein mungkin. Jadi alternatifnya kalau memang oksidasinya terlalu berlebihan, bisa ditambahkan antioksidan untuk mereduce dampak dari teknologi plasma ini. From Ibu Lilis, is there any possibility that plasma treated water with nitrite content of 100 ppm commercialized in bottles? So we can buy it from supermarket and we can use it at home. And is it considered as food additive?
Well, well, it's the illegal issue is different by government Well, so if your government or my government say this is food additive, then it is food additive. But actually, we did not add anything, right? We have water and plasma treated to water, and you put into the powder, then sell it. So yeah, so it's a legal fact.
Seperti itu Bu Lilis, mungkin ya tergantung kebijakan dari pemerintah negaranya masing-masing, dan memang ini diperlukan referensi-referensi yang banyak, jadi nggak cuma Projo doang yang mengerjakan ini, bisa dibuktikan nanti sama Bu Lilis di sini gitu ya, apakah memang dia bisa dikomersialisasikan sebagai food additive atau nggak usah di label food additive jadi air bernitrit gitu ya.
Because I think the questioner say that, well, irradiation is approved as food additive, right?
Okay.
Yeah, but the irradiation is not add anything to food.
Yeah,
so maybe plasma could be food additive in some government or circumstances, but it's not actually add something.
Jadi kalau iradiasi contohnya itu masuknya labelnya food additive. Kalau ya nanti mungkin Bu Lilis bisa mengembangin teknologi ini, Mungkin nanti bisa minta tolong ke BPOM ya, melegalkan dianggap sebagai food additive juga.
Oke, terima kasih. Thank you, Mr. Joe.
Next question from Ibu Putri. It's so interesting topic about cold plasma technology, and what do you think whether this technology could combine with omics? And how about from the nutritional value, such as essential fatty acid, amino acids, and so on? Is there any change or significantly increase?
Well, I don't think the plasma technology change all any nutritional values. These macromolecules like proteins, fat, or others. Was maybe some amino acids, not amino acids, some vitamins like vitamin C or vitamin E could affect, because it is easily oxidized from some oxidative materials. But usually the macromolecules, nutrients could not be changed by this small energy of plasma. Okay, and it is very interesting topic to go to the omics technology, because some of researchers, not me, but some of researchers found that if you put the plasma, very low plasma energy to plant seed, then those plants grow faster, bigger, and stronger. Okay, and one of the researcher also did in egg, poultry egg, chicken eggs, and they found some DNA methylation story, and they have a bigger growth or a faster growth of chicken, right? So may have some effect on or DNA. I'm not sure, but they have some researches on this technology. But as I said, for macromolecules, I don't think any changes in nutrients or fatty acids, whatever.
Oke, seperti itu Bu Putri. Mungkin akan terbuka juga untuk Bu Putri membuktikan ya di bidang pangan, mungkin dari produk pertanian dan juga peternakan. Mangga kerjasama sama saya dan juga Prof. Jo untuk membuktikannya bareng-bareng. Next question from Pak Diki Ramdhani. I'm currently work with plant bioactive compounds as natural additives and anthelmintic in ruminant diets to replace antibiotic growth promoter. His question, do plasma treatment can help to improve the quality of the bioactive compounds? Second, the cost of plasma treatment reliable for feed industry.
Okay, this is also a good question. Well, I did not explain about this biological activity with plasma for this today's presentation, but if you use the plasma in certain type of bioactive compounds, then there is some chemical reactions occurred with and produce some different kinds of products, and those products, some of those products have a lot stronger, for example, bioactivities like antioxidant or anti-diabetic activity, all those things. Well, there are some research found those kind of thing. And also the cost, well, if you could find, well, there are a lot of different companies that could install those plasma operator. If you find those but what can I say, this manufacturer, then for plasma treatment, you can use only electricity. So, well, it could be reliable. in terms of the cost.
In terms of the cost, sangat reliable ya Pak Diki. Mungkin bisa dicoba ya di sini. Kita bareng-bareng coba. Kemudian tadi ya kita buktikan juga yang terhadap berbagai macam bioactive compounds, kan Prof. Joe juga tidak menceknya sendiri, jadi mungkin butuh data juga dari Pak Diki nanti di masa depan. Kita collect bareng-bareng. Kemudian dari Irawan, from the economic point of view, how is the simple comparison of processing costs using cold plasma with conventional method?
Well, actually, actually I did not calculate the economic cost exactly, because some of industry interested in this plasma technology, but they did not really committed to build as a commercial status, because they have to have consult with the food safety authorities, Korean Food and Drug Administration, about legally approve this process. Okay, so it is quite complicated. So there is no commercially available evidence for give you exact amount of economic points. Sorry about that.
Oke, I think Pak Irawan juga bisa berkolaborasi nih memecahkan masalah ini. Ya mungkin ya karena di Korea juga regulasinya belum kelar, jadi memang sampai ke economic point of view-nya masih belum perfect gitu. Kita Prof. Joe masih mencoba di level pilot scale mungkin ya di dalam lab. Kemudian dari from Ibu Novi Mayasari, first question, have you studied risk benefit? Again, same question, what is the big challenges? And is there any types of plasma?
Let's see. What is big challenge of this technology? I think the challenge is approval for consumer. I mean, the approval of this process from the authority. Okay. Irradiation also very important and also very safe processing technology. However, some countries did not approve those irradiation technology as a food treatment, right? As like this, the plasma also new technology, and need to get a legal approval, which is time-consuming process. That could be a challenge for the industry. And how many types of plasma? Well, I can say plasma is plasma, and there are different type of plasma which have different advantages, like DVD plasma, pen-type plasma, or arc plasma. Well, those plasma type could be developed as your purpose, and those plasma system could be very easily understood by physicians, okay, majoring in physics, plasma physics, or some of engineering part. They understand very well about plasma system. So if you can have some connection with the physics or engineering part with plasma, then you can have a very, very efficient and easy way of research can be done. I really recommend to work together.
Thank you, Prof. Joe. Ya, Bu Novi udah diajak tuh. Jadi memang Prof. Jo juga berkolaborasi dengan Korea Atomic Research Center dan juga orang-orang dari fisika. Jadi untuk mengaplikasikan ini di pengolahan daging. Jadi beliau juga belajar dasar-dasarnya. Bahkan untuk mendesain mesinnya, alatnya juga butuh bantuan orang teknik gitu ya. Jadi emang nggak bisa sendiri. Jadi kalau ada di sini mungkin siapa aja bisa berkolaborasi.
We are waiting for the collaboration in the future. Yeah. Thank you so much for the answer. Yeah.
Next question from Kimberly. Since plasma can oxidize fat, then is plasma treatment limited to fatless meat?
Okay, I understand. Well, I don't think so, because even though there is some fats, the level of oxidation or the level of that people actually feel is important. All right. So even though you have a fat in food, you can use plasma if it is okay for people. So there is some test to have any sensory change before you use the plasma technology.
Okay, thank you. So Kimberly, ini kembali lagi ya, tujuan plasma awalnya itu hanya untuk microbial inactivation, kemudian berkembang ke manfaatnya kalau di pengolahan daging, menggantikan curing menggunakan nitrit yang ditambahkan secara nitrit sintetik yang ditambahkan. Jadi sebenarnya plasma sebagai antimikroba itu bisa aja dipakai untuk segala jenis bahan makanan yang mengandung lemak. Namun kalau untuk minyak sendiri ya, minyak kan itu udah lemak semua, kayaknya nggak perlu ditreat pakai plasma juga, karena kan tidak terlalu beresiko dengan mikroba patogen. And then, again from Sophie, if I want to research about poultry further processing without additives, do I have to go to your research facility in Korea or any plasma research center? I think
always welcome.
She wants to be your student.
Well, always welcome, but it is too far from your state, your place to here. As I said before, maybe you can develop some plasma system with your co-workers, like physics or engineering departments, right? Also, Thailand have a very good plasma research group, as I understand. Okay, Thailand, and also, well, but still, if you want to come to here, well, I welcome.
Okay.
Thank you, Professor. This is a long-time project for non-additive poultry. Thank you.
Okay, thank you.
Sophie. Mangga, Bu, kalau ingin kolaborasi dengan beliau, berkunjung ke sana, mangga. Kemudian kita kembangkan di sini. Tadi kan beliau referensinya Thailand tuh udah start, ya kita bisa start juga di sini.
Boleh tahu Thailandnya di mana ya, Pak? May I know the Thailand plasma research?
Julius, do you know where exactly the Thailand plasma research center?
Let me see.
Is it university?
Yeah, one of the university, and well, I can send you the affiliation, but also you can find cold plasma and research Thailand. Then you can find a lot of not too many, but some papers from there, right?
Thank you. Ya, mungkin Google nanti bisa membantu, Bu.
Thank you, thank you.
Oke, apakah rekaman ini dapat diakses kembali? Oke, nanti akan saya siapkan. Terima kasih. Oke, no more question. Is there anyone yang mau bertanya secara langsung? Mangga. atau terkait lainnya, kerjasama ataupun studi.
Yeah,
Jika
maybe
tidak,
is it okay if I say
Bu
something?
Novi, mangga.
Okay, thank you, Dr. Diki. Hello, Prof. Joe. Thank you so much for very insightful presentation. It's really new actually for me because I work from different field, still in livestock, but it's really, yeah, I see a lot of opportunity to work together, I think in collaboration because, yeah, basically I'm from Department of Nutrition and Feed Technology. So basically we also have some requests from industry to find a good way to replace synthetic compound to keep the meat healthy and also more organic way and things. So we really hope that we can have a future collaboration for research and also probably in education for summer course or for other occasion. So I would like to say thank you so much, and I hope this is not the last one. So this is the first, but then we can continue for another collaboration. I think that's all from me. Thank you so much. Yeah.
Thank you.
Thank
It was
you.
very interesting discussion too for me.
Yeah.
Okay, thank you, Bu Novi. Thank you, Professor Joe. I think we have to finish this lecture.
But sometime later, I'm sorry, Dr. Dickey. Sometime later, I really would like to go to your place and actually see in reality after this
Yes,
COVID-19.
please. Yes, please.
We have golf jang here. We can play together.
Maybe we can, Pak Diki, maybe we can also make like a joint supervisor, something like that. Or maybe we can
possible.
also have write together, something like that.
And
So,
I also have a plan for next year, maybe research collaboration with him. So we can start as soon as possible. No waste of time anymore.
yeah, just make a plan for that.
Okay. Okay. My apologize. We have, as we have time limit. I am very sure it's already lunchtime. So I'm really sorry. I should end the lecture. On behalf of faculty members, I would like to invite Dr. Lilis Suryaningsih as coordinator of this course. Ibu Lilis, Bu Lilis akan menyerahkan sertifikat apresiasi kepada Prof. Joe. Panitia siap menampilkan sertifikatnya. Oke, mangga Bu.
Yeah, okay. Thank you, Professor Joe, for your interesting lecture. Yes, I think this lecture is very useful and helpful for all of us. Thank you so much. Come back to next year.
Kamsahamnida bahasa Korea.
Makan di Mujigae. Okay.
Thank you. Thank you very much.
Okay. In Indonesia, restaurant Korea is very famous, Mujigae, Mr. Joe.
We have a popular Korean restaurant here already. So Korean food already invade us.
Okay.
Joe's name again, Kamsahamnida. I will see you soon, hopefully. Terima kasih Bapak, Ibu, dan para mahasiswa atas antusiasmenya, atas partisipasinya bagi peserta mata kuliah PPD. Jangan lupa konfirmasi kehadiran ya di akun chat masing-masing. Sampai jumpa kembali di kegiatan sharing ilmu berikutnya ya. Ditunggu Salam sehat dari Jatinangor. Saya mohon maaf undur diri. Wassalamualaikum warahmatullahi wabarakatuh. Thank you and have a good day.
Waalaikumsalam.
Thank you.
Terima kasih Pak
Thank
Diki,
Thank
Pak
you,
Diki,
Prof.
Kamsahamnida,
Professor
Prof.
Thank you.
Jo. Ya.
Okay, I'm going out.
Okay.
Oke, have a nice day.
You too. Thank you.
Thank you.
Bapak
Terima
Ibu,
kasih teman-teman
ya.
upgraders, terima kasih ya. Pak Diki terima kasih.